Hello everyone, we are back with our new chapter, which is chapter number 21, and which is on the topic of biotechnology. Right, so let us begin. I'm sorry for that noise. I actually live next to a highway, so it's always very noisy. So let's begin with the genetic engineering definition, which is changing the genetic material of an organism by removing, changing, or inserting individual genes. So for this chapter, there are not much definitions to remember, but there are certain processes that you guys need to remember, and it's mainly about bacteria and cells actually. So let's move on, which is exercise 21.1, pectinase. In this exercise, you are asked to think about the best way of displaying a set of data to emphasize a particular point. There is no single right answer, so try out 2 or 3 different ideas before finally constructing your graph or chart. So pectinase is an enzyme that is used in the manufacture of fruit juice. Pectinase is produced by growing microorganisms such as Penicillium vidiricatum in fermenters. So let's move on to part A. List 3 reasons why microorganisms are often used for the production of enzymes. So this is a very important question, a very popular question that almost comes out every year. It's not almost every year, in every season actually. So always remember these 3, at least 3. Minimum they usually ask about like 2 marks question, but it's best to memorize about like 3 points. So they're very small, and therefore they're easy to grow in large numbers. This itself is actually 2 marks. First, they'll ask you to explain 1 point and how does it actually affect it, and then another point you could explain there are no ethical issues associated with their use. Another point, there are no ethical issues, which is 1 mark. And then they have the same type of genetic material as other organisms. So this statement is very common in MCQs, like literally very common. This and this, the ethical issues and the same genetic material. It's a very popular MCQ question for this chapter. So take note of this. So let's move on to part B. What kingdom does penicillin belong to? So you guys have to remember there are actually 5 kingdoms. So among them is fungi. So name a useful product that is made by different species of penicillium. So a useful product means a product that's that has a good benefit actually. So you could say it produces antibiotic, and antibiotic, another scientific name is penicillin, right? It's actually from the species Penicillium, but it's also known as penicillin as well, like from where it's derived from. But a common answer that you guys have to normally write is antibiotic. Right, so part D. Right, so the table shows the production of pectinase by P. vidiricatum, which is grown on different kinds of waste materials from food processing. Bagasse is the waste material left over from the processing sugarcane. So this is the table. These are the substrate types and production of pectinase in Abiturian units. Right, so just take a look at all this. Now, moving on to this is display these data on the grid on the next page in a way that clearly shows how adding sugarcane bagasse to the substrate affects the production of pectinase. So now, over here, if you see, they didn't really specify what grid you guys have to draw. But looking at this, and looking at the table, what they've given you, substrate and the production of the pectinase, it's a very common, like you guys may commonly think that, oh, we might have to draw a line graph, because it's actually, they usually ask that in paper 6, so we have to definitely draw a line graph. But actually, you guys must bear in mind, for drawing a line graph, both the x-axis and the y-axis must be both in numericals. There must be in values. Like both the x and y-axis must have numbers. But over here, if you see, one set of data is in words and another set of data is actually in numbers. So in this case, the only suitable one that you guys can draw is actually bar chart, right? So I'll show you guys a picture of this thing, and right, so this is how the bar chart looks like. I actually didn't have like a proper graph paper, so I actually drew it on this paper, but you guys, since a graph is actually already provided to you on the question paper, so you don't need to separately have it. But for my case, I didn't have it, so I just did it on a normal piece of paper. So this is how it looks like. So as you can see, it increases and then it decreases. It's more of like it's very discontinuous actually. So bear in mind that you need to remember there's actually a rule for drawing bar chart, and that rule is there must be equal spacing between the charts. But as you can see, for my case, I don't have equal spacing because since it's not a proper chart, so it's not possible to fit everything in one plane in equal spacing actually. And but for your case, you guys have to make sure that the spacing between the charts, they're actually equal. If it's like 3 boxes, or for example if it's 10 boxes, like on your graph it's like 10 boxes, must be 10 boxes between all these 3 bars. But if it's like 5 boxes, must be equal spacing, 5 boxes 5. It's a very general thing you guys must be knowing. And yeah, so the substrate will fall on the x-axis and the numericals will fall on the y-axis, right? So okay, so let's move on. Right, so suggest how using food processing waste materials as substrates for the production of pectinase could benefit the environment. So let's read out the answer together. If the waste materials are not used, then they have to be disposed of, right? So they might pollute waterways, causing eutrophication, as they are likely to contain nutrients that could be used by bacteria, which would then use up a lot of oxygen as their increased populations respire. Also, by not using waste materials, the plant material would have to be used to make the pectinase, which means more land would have to be used for growing plants that might otherwise be used for growing food or for habitat for wildlife. Another reason it decreases the variety of the species of the plants, as not all plants can be used for making pectinase actually. So it depends. So basically it's two points you have to state over here. The first reason is what would happen if the waste materials are not used. So it could pollute waterways. This is like the first point, it could pollute waterways, right? And the second point is plant material had to be used to make the pectinase, which means more land would have to be used for growing the plants. This is the second point, right? So explain why pectinase is used in the manufacture of fruit juice. So pectinase breaks down the pectin that holds together the cells in the fruit. So pectinase is actually an enzyme that breaks down pectin that is present in the cells of a fruit. Example of fruit is apple, right? So this makes it faster and easier to extract the juice, and also increases the total yield of juice from a given mass of fruit. Pectinase might also make cloudy fruit juices clear. So there are actually like 3 reasons why pectinase is used in the manufacture of fruit juice. So normally when you make fruit juices at home, it's usually very cloudy. It's usually very juicy type. Not juicy. It's very thick. It's I mean, the juice that you might that you guys might make at home from using just crushed apples, or by using crushed apples or oranges or mangoes, you won't be getting that much pulp in your juice if you don't use the enzyme called pectinase. So with the help of pectinase, it actually helps the pulp, like more pulp is actually is present in your juice. So you don't really write pulp in the question paper, what you write is it increases the total yield of the juice, and it makes the extraction faster and easier. And another point, it makes cloudy fruit juices clear. So if you see in supermarkets, if you buy these packet fruit juices, they're always clear. You will never find like very cloudy-ish feeling, a very thick feeling when you're consuming fruit juices that are purchased from the market. That is, and the reason behind that is because they use this enzyme called pectinase. So yeah, that's it. And moving on to the next part, this is the graph. I've shown you guys the picture earlier. Now moving on, the next part is exercise 21.2, which is on yogurt. So let's read the text. You don't have to know how you have to make yogurt, but it's this exercise is mainly about understanding complex data, analyzing the data, drawing conclusions from them, and relating them to a real life situation. So it's mainly on pH and the effect on separation. So let's read this out together. Yogurt is an important food in Pakistan. Yogurt is made by adding particular types of bacteria to milk. The bacteria changes sugars in the milk to lactic acid. Until recently, most yogurt was made and consumed fairly locally. Now yogurt is being made commercially on a larger scale, which means it has to be transported over larger distances and kept for a longer time before it is eaten. The yogurt makers therefore add stabilizers to the yogurt. Stabilizers help to stop the yogurt separating out into curds, the semi-solid material, and whey. So basically, when you make basically it's an artificial yogurt, you should say. Artificial in the sense that they add stabilizers to make sure that the yogurt doesn't spoil very fast. Whereas if you make yogurt at home, I'm not sure if you guys make that at home, but if you make actually fresh yogurt at home, after a few days, after being kept in the fridge, even after being kept in the fridge, it will actually start separating. It will start giving out water from the yogurt. It's completely normal. It starts giving out water, and people generally don't like seeing water in yogurt as it makes the appearance seem too bad. So what these commercial manufacturers do, they add stabilizers to make sure that the yogurt does not separate out into layers. Right, so they're actually, so let's read the last sentence, which is the effects of 3 different stabilizers on yogurt made from buffalo milk were compared. The tables show some of the results. Table 1. Effect on pH. The numbers in the table are the pH of the yogurt. So this is a stabilizer. When there's no stabilizer added, the pH of 4.1, it dropped over the day of 15. But when you use stabilizer pectin, it was initially they were all 4.1 the pH, but over time it gradually reduced as well, but it reduced very slowly. I mean, I wouldn't say slowly, but it was fairly still okay. And the stabilizer carrageenan, this one actually reduced like really slowly, 4.1, 4.1, 3.8, 3.9, compared to this one. It's like a 0.3 difference, and then there's like 0.1, and then yeah. But this carrageenan, the pH actually remained the same on day 5 as well, but on day 10 it actually reduced. And then the stabilizer cornstarch, it reduced to 4.0 on day 5. On day 10 it reduced to 3.8, and then it again increased back to 4.0. Now table 2, the effect on separation. The numbers in the table are the degree of separation of the yogurt. The larger the number, the greater the separation. Right, so if you see we're here, stabilizer 1.9, 7.9, you see there's a huge difference. That means definitely it has separated. 0.9, 2.5, definitely it has separated. This one, 2.3, it has also separated. And cornstarch, it remained fairly the same. I mean, not same, but it wasn't that much difference between if you compare this row with this one or this one. So now let's move on to the first. So before I actually move on, I will actually make a second part of video on this. So stay tuned, and I'll be putting the second part of the video very soon. Alright.